My name is Masato Kubo. I'm in charge of the Division of the Molecular Pathology at Tokyo University of Sciences Research Institute for the Biomedical Sciences, which we call LIBS for the short. First, I want to tell you a little bit about the LIBS. This institute was established 25 years ago. We focus on the basic research in the medical sciences. LIBS has been playing an important role in helping basic medical science move from the lab to the real world, such as into the clinical sciences and drug development. Next, I will introduce the division of the molecular pathology. The division mainly focuses on the cytokine, which are secreted protein released by the cell that has a specific effect on interaction and communication between the cells. We have been trying to understand the role of the cytokine in the host defense and the numerous diseases caused by the immunological responses. We are working on the two major subjects: vaccinations against the influenza infections and allergic disease, including asthma and atopic dermatitis. Today, I would like to introduce some recent challenges related to these two issues. First, I will talk about some influenza issues. The spread of the flu virus is a serious public health concern. It has caused several pandemics in the past few years. Vaccination by inducing antibody have been important in helping to prevent the spread of the flu. However, no vaccine offers enough protective antibody against the emerging viruses. This is because the effectiveness of current vaccine rely on the influenza strain-specific immune responses. So, a better understanding of how antibody block a virus infection is need to develop alternative vaccines against the potential emerging pandemic viruses. It is virtually textbook dogma that neutralizing antibody induced by antivirus vaccine requires a specific kind of T cell called the TFH for the antibody responses. Our recent research has demonstrated that different kind of the T cell, Th1 cell, also produce a cytokine called the interferon gamma that trigger the production of protective antibody response against the flu virus. Therefore, we believe that these Th1 cell could be a promising strategy to induce an antibody that can neutralize the influenza viruses. Now I will talk about issue related to our work on allergic asthma. Allergic asthma is a chronic inflammatory disease that can cause the airway to tighten and cause wheezing. Conventional asthma model have shown the importance of the specific type of the T cell called the Th2 cell in controlling this type of the allergic response. Most recently, our research have shown the importance of IL-4. We have found that this cytokine secreted from a basophil, a type of the immune cell, is a key to triggering asthmatic inflammation, such as that induced by the house dust mite. I hope that our work will continue to shed light on how cytokine control the immune response to several kinds of the disease. Someday, I hope that our research can form the basis for the cytokine target therapy, creating a new medicine or treatment to cure or stop the disease.